As we continue talking about viruses, we're going to kind of focus in on bacteriophages. They're a great example for us to learn about how viruses infect their host and how those infection cycles can take place. So we talk about this particular bacteriophage. They have a really complex capsid, and they actually use their capsid to inject their DNA into their host. Now, once a bacteria gets their nucleic acid into the host, that's when the overall viral genome is going to take over and start manufacturing viral proteins. So the virus actually uses host enzymes, host ribosomes, tRNAs, amino acids, the host ATP, all the host different mechanical systems that are inside the cell. The virus hijacks that. And that's one of the reasons that we previously talked about host range and how viruses can have a pretty narrow host range sometimes because they need to be able to have the cell make their next generation. Now, viral nucleic acids molecule and the capsomeres, they're actually going to spontaneously self-assemble. So they don't need a whole lot of control elements inside the cell. They just need to be produced. And so once that genetic material is in there, the virus is going to take over the cell and produce a whole lot. So what are we talking about here? So here we have kind of a virus that's coming in and it's going to infect a cell. So a virus comes into a host, and so its actual capsid is going to break down. In this case, it's got a viral DNA. It can go one of two directions. DNA polymerases can actually make new viral DNA, and they will. Or we can see transcription occur, and viral mRNA will get made that then is used to make these capsid proteins. Now that we have both of these two different things happening, we end up self-assembling new viruses and moving on. Now, this is a general overview of a virus infecting a prokaryotic cell. So let's talk about the different stages that these phages can actually undergo and what's going to happen. So we understand phages the best because we've been able to study them the most. And they basically have two different reproductive mechanisms, a lytic cycle and a lysogenic cycle. Now, the lytic cycle is a phage replicative cycle that culminates in the death of the host cell. So you can think lytic cycle, you think it's going to lyse or split or go through lysis to break up that cell. And so the lytic cycle produces new phages and lyses or breaks open the host cells, releasing the progeny viruses for the next generation. The phage that reproduces only by the lytic cycle is typically called a virulent phage. It's never going to be dormant. It's always causing an infection. Bacteria have actually cooked up defenses for this called restriction enzymes. And we'll continue talking about some of our uses for restriction enzymes in the biotech unit. But restriction enzymes basically recognize and cut up DNA. So you can see the defense system here. These bacteriophages get their genetic material inside of the bacteria, but the bacteria has an enzyme there that's recognizing it and chopping it up to stop it. So how does that work? So here we see a T4 bacteriophage. And what the T4 bacteriophage is going to do is actually land on the surface of a bacteria and inject its genetic material. The protein doesn't actually go into the cell. So when we see this third stage here, it says synthesis of viral genomes and proteins, those viral proteins didn't enter the cell. They were created in the cell in the manner we talked about previously. They're going to spontaneously form into the new next generation. They don't need any kind of mechanism to help them do that. And so they're going to get assembled inside the cell. When that occurs and enough of them get made, the cell is kind of bogged down. It's been abused and overused, and it actually is going to bust open and release all these cells. So in a way, these lytic cycles are just little ticking time bombs waiting to explode more cells for the next, or explode onto the other cells, all these new viruses for the next generation. The lysogenic cycle is a little bit different because in this particular one, the phage genome doesn't actually destroy the host. So the viral DNA molecule is incorporated into the host cell's chromosomes. This integrated viral DNA is sometimes what we call a prophage, and every time the host divides, it actually copies the virus itself. An environmental signal can actually trigger the virus genome to exit the bacterial chromosome and kind of kick into a lytic mode. So phages that use both the lytic and lysogenic cycles are called temperate phages. Now, if a phage were to only use the lysogenic cycle, it would get stuck in the genome and not go anywhere. So we basically get two types, lytic viruses and temperate phages that are going to use both. And so what the phage now has in this setting is once it gets its chromosome into the bacteria, it's either going to go straight to lytic, be virulent, be this destructive to the host kind of thing, or it's going to kick into the lysogenic phage and it's going to incorporate its genome. It gets replicated and passed on, and there's some other event that's going to kick it open. So when we look at this whole picture, we can kind of see it forms like a kind of figure eight. And we start up here in the upper right. This is the phage that lands, injects its DNA. This is the point where we had on the previous slide, where right now we have kind of a fork in the road. Typically speaking, with these viruses that are going to be those temperate type viruses, they're going to kick into the lysogenic cycle for a little bit. They're going to include their genetic material into the host. The host will replicate a bit, and then later on, something's going to kick them back into this portion. Some kind of environmental, some prophage is going to exit the bacterial chromosome, and now it kicks into the lytic cycle. Here in this new cell that has been dormant for a while, potentially, you're going to go right back to that lytic kind of cycle, where what we're going to do is generate new copies of the nucleic acid, new copies of the capsids, and we're going to produce new fully formed bacteriophages, and then they end up bursting the cell, spewing them all onto their neighbors and causing a large amount of problems. So this is the whole picture of that lytic and lysogenic cycle. And one of the things I don't like about these particular examples is the way they're named. Because in the lytic cycle, we said, oh, remember, that's going to lyse the cell. But then we have a cycle called the lysogenic cycle. It's not my favorite naming that happened here because it gets confusing. But in the lytic cycle, that's where lysis actually occurs. In the lysogenic cycle, this is where it's going to be dormant and you're not going to see it, at least for a short period of time. So again, here's lytic. We're going to see those cells rupture. That's lysis. That's the cell lyses, releasing the phages. In the lysogenic cycle, the bacteria actually reproduces. The prophage is formed. And so those are the two major ways that we see bacteriophage life cycle in cells.